All right. Hi, everyone. My name is McLaughlin, and I'm a PhD in neuroscience from the University of Pennsylvania. And my research focused on mapping out how a specific neuronal circuit, right in the middle of the brain, very deep under the outer layers, how it regulates anxiety. And it's a circuit we've known has been involved in the horrible syndrome that people experience after they quit taking various drugs, from alcohol and nicotine to cocaine and heroin. But my work was focused on whether this particular constellation of neurons connected to other neurons regulates anxiety independently of drug withdrawal. Well, I just wrapped up some of the last revisions of the paper that describes my sort of final set of experiments that I did as a PhD student. It's sort of like a diary of all the discoveries we made as a team. But because the story isn't completely finished, I'm almost certainly going to be handing the story off to a new author who will write the final chapters. And, scientific publications are, of course, fundamentally different relative to something like a diary or a journal, but there's a similar feeling of sort of personal investment because, after all, I'd spent the 14 to 16 hour days performing viral infusions or immunohistochemistry to get the story to its current state. And, walking home at like 4 or 5 a.m. just hoping that an experiment was going to work out. So it's sort of impossible to avoid that sense of like personal investment in the direction that the story takes. But that's also sort of the beauty of science. It doesn't really matter who's writing the story. It's the discoveries and maybe more importantly, the unforeseen opportunities for new discoveries that determine the trajectory of a scientific effort. And of course, that's the whole point of peer review, to extract any possible influence of this kind of personal, emotional investment in research. But regardless, to be perfectly honest, it's still kind of tough to hand this project off to somebody else. Anyways, all right, thanks for indulging my sort of bittersweet eulogy for my time with this project. And it's going to be in the hands of, some new grad students who will, hopefully be ambitious enough to see it through to its conclusion. Okay, well, while I'm still planning on taking a bit of a hiatus from COVID-19 related stories, particularly since there haven't been really many meaningful findings that change what we've already discussed on past Sundays, I thought I might talk about an article that was published by the science writer Carl Zimmer in National Geographic about the role that viruses have played in devising what ultimately has become our genome. And I'll go a bit beyond the article as well, but it's a great story. So, and I should note, before I totally depart from COVID-19, there was a study that was published in the journal Brain that corroborates, some of what we already discussed in terms of the neurological symptoms manifesting in some people who become infected with SARS-CoV-2, causing COVID-19. And, once again, the study notes that there were some patients who didn't present really any of the classic symptoms of COVID-19, and it was just the presence of neurological symptoms that indicated something was wrong. But there was a new set of symptoms that was described in this study, and the study was conducted in the UK. So 9 of the patients in the study developed acute demyelinating encephalomyelitis. So that's basically where the immune system seems to be attacking myelin cells. And so this is, sort of similar to multiple sclerosis or Guillain-Barré syndrome. This syndrome is pretty rare, and more worryingly, it doesn't tend to happen in adults as frequently as it does in children, but in this study, it was adults who exhibited this symptom. And, one patient report was pretty concerning. So it was a woman in her mid-50s who had no history of psychiatric conditions and who was treated for COVID-19. And, she was exhibiting the classic symptoms like a high fever, coughing, muscle pain. And she was treated with oxygen and discharged from the hospital after about 2 weeks of treatment. But several days after getting home, her family reported that she was having bouts of psychosis. And, severe psychosis, like hallucinating various animals like monkeys and stuff in the house that weren't there, and exhibiting confusion and sort of odd aggression. And the study doesn't describe the fate of this person. Perhaps we don't know. It's still sort of an ongoing situation, but it's just another data point suggesting that the ramifications of COVID-19 are likely broader and potentially even more severe than I think is kind of widely appreciated. And to be clear, this isn't a super recent. So that publication was quite recent. It was in the past few days. So I just thought I'd bring it up just to sort of corroborate this, the presence of neurological symptoms in some people who develop COVID-19. So done with COVID-19 for now, at least for now. But we are going to talk about viruses. And I should say, this whole sort of story about the relationship between viruses and our genome, it's not like a particularly new story, but some of the discoveries that we'll discuss were published like maybe 2 years ago or so. But then, some of the research extends far back. I think it's an interesting part of the story of human evolution and just like evolution in general. And I don't think it's like widely understood or appreciated, this symbiotic relationship with some viruses. Okay, so what is this relationship that humans have with viruses over the course of our evolution? So I have to imagine that almost without exception, most people would assume that viruses are exclusively dangerous little things that invade a biological system and either ultimately destroy it or succumb to destruction by the host's immune system, regardless of the animal, particularly these days. And very frequently that is the case. When it comes to things like the influenza virus, which causes the flu, that's certainly the case. And of course, the virus thrives by being able to spread before one or the other happens, before the virus dies or before the host animal dies. But occasionally, throughout history, there have been examples of viruses that have integrated into the genomes of the hosts that they infect. And more interestingly, though, in these rare cases, and they are rare, the viruses have ultimately been passed down through the progeny of their hosts, essentially like becoming a part of that host's genetic lineage. And, we can tell when this happens by sequencing human genomes. And as it turns out, viruses have pretty distinctive genetic signatures that, distinguish them from animal genomes. And so, while viruses don't leave behind traces of their existence in like fossils, like we may be used to, they do leave a kind of fossil record in the genomes of the animals they've infected in this way, by being integrated into their genomes. And so one example of this kind of like fossilized virus nestled within our genome is they're particularly old sequences in our genome, sequences derived from a viral infection, tens of millions of years ago, and that, because they've been there for so long in the genome as it's evolved, they've accumulated a lot of mutations, and those mutations changed the sequences from their original version, such that they don't really do all that much of anything other than just preserve the record of that initial infection. And so it's sort of like you can imagine like a fossilized T-Rex can't like chase you, while you're driving away in a Jeep, but it does demonstrate what that original version of the dinosaur looked like, how it hunted, and other things we can learn from just like looking at the fossils alone. But if viral sequences are integrated into an animal's genome, that will tend to slow down how rapidly that virus would otherwise mutate. So, basically like those sequences will only mutate as frequently as the genome of the animal mutates. So, relative to how, rapidly many viruses mutate and evolve, like the influenza virus notoriously rapidly mutates, once it's integrated into an animal's genome, it sort of preserves its sequence, right, in time, and sort of preserves it for a lot longer than it would be otherwise. So, viruses, just so we're all on the same page, they're just very simple sets of genetic instructions that arrive in usually a super microscopic delivery system, right? They're just DNA or RNA, just like the DNA or RNA that's being read, created, and translated into stuff that composes you right now at this very moment. And it's typically encased in some kind of package that enables it to sneak into a cell of some sort to trick the cell's machinery into creating more of the virus. That's the story we're used to. But they're really rather simple relative to the organisms that they infect, particularly if you focus on like animal hosts. So as an example, SARS-CoV-2, the virus that causes COVID-19, has about 30,000 RNA bases, about 15 genes. The human genome has about 3 billion base pairs and between 20 to 25,000 protein coding genes. So, even a virus that's basically brought many countries to their knees is composed of a genetic sequence that's like a fraction of a fraction in length of the genetic instructions that serve as the blueprint to make a human. So, by the way, not all viruses are super duper tiny. I love this little subset of research. There's a whole group of viruses known as giant viruses or gyruses. And they're sometimes even larger than bacteria, hence the name giant virus. And so here are some examples of what they look like. So at the top, the top picture is a mimivirus. followed by a megavirus, followed by CRO-V. So these are 3 different types of giant viruses, and they have gigantic genomes relative to other viruses, like closer to 1,000 genes rather than like 15. And so the whole story about giant viruses is super fascinating, and part of the reason that the lines between what we consider to qualify as life and otherwise begin to blur, right? The debate as to whether or not viruses should be considered living organisms. And by the way, I should note that new versions of these giant viruses are discovered like more frequently than you would expect, more frequently than I'd expected at least. Like when people go out into nature, into like areas where there's water or whatever, new habitats, they'll find these giant viruses. And so on the top there, that the Mimi virus, that was the first giant virus that was discovered, and it is like huge, it like 750 nanometers across. And so some scientists hypothesize that viruses like these giant viruses may be the byproduct of the evolution of other, even bigger viruses that perhaps, even more closely resembled living cells, but ultimately evolved to just exploit the machinery of the living cells that were around them instead of taking the cell route. So anyways, given that these viruses tend to have these short stretches of base pairs, DNA or RNA, A, T, C's and G's, or A, U, C's and G's in RNA, and that they tend to just be a handful of genes, they can be pretty easily identified when geneticists know what to look for when they're when they're intentionally looking for viral sequences. So within the human genome, geneticists have come across parts of the sequence that looks sort of virusy, right? So according to Carl Zimmer, the ones that were the easiest to identify were the retroviruses, and you're almost certainly familiar with one particularly infamous retrovirus, HIV. So this is the virus that causes AIDS and or acquired immunodeficiency syndrome. So it's a type of virus that has sequences that enable it to not only infect cells in the body, but also insert copies of its own genes into the host's genome, right? That's why they're called retroviruses. And so as a result, they're called retroviruses due to the mechanism by which that happens. But all retroviruses have some capability like that. And so as a result, the host's cellular machinery is reading through its genome, like is happening in your body right now, and it just goes ahead and copies the viral genes along with its own. And so this then produces new viral particles, and then the cascade continues, in the way that we're used to, right? And this is what generally happens. But every now and then, over the course of history, a retrovirus can end up modifying the genomes of germ cells, very important and special cells. These are egg cells or sperm cells. And so, if you're listening to this or watching this, you almost certainly are aware of the roles that eggs and sperm play in making a baby, so we won't get into that. But the important part is that if the genomes of eggs or sperm are altered, that means that basically all of the genes of a potential product of the participation of that egg or sperm cell in creating a new human will also be similarly altered. So those viruses go from being like exogenous retroviruses to endogenous retroviruses, and we call them accordingly. We call them human endogenous retroviruses, or HERVs. And so this ability to modify an organism's genome way off into the future, forever, it's kind of similar to the concern regarding the possible use of a genome-altering technology like CRISPR-Cas9 to modify embryos at the earliest possible stages. Those genetic changes will basically persist throughout the rest of the resulting person's progeny. It's not like it's just a chance. Like, the genetic changes at that level can have a permanent influence over that human's genetics, as well as potentially the genetics of many other humans over time. And so, CRISPR was developed in a lab and can be, it's derived from an actual natural, wild biological mechanism, but it was ultimately developed in a lab and can be used to modify a human's genome in the span of just like a few months or years. And so these viruses that we're talking about here, these retroviruses that became integrated into the genomes of animals, they evolved over far, far longer stretches of time, right? So if one of these viruses that can integrate into its host genome finds its way into a germ cell, sperm or an egg cell, it can essentially piggyback on the evolution of the host. And that's what happened in our own genome. So we carry about 100,000 pieces of DNA derived from retroviruses. According to Zimmer, about 5 to 8% of the human genome is composed of the sequences derived from retroviruses. And so this isn't like unique to humans alone, right? It takes many years, millions of years, for the accumulation of all these virus-derived sequences to occur in organisms. And while I said that some of these like fossils of viruses present in our genome aren't particularly functional anymore, that's not always the case. So for example, some of the very same viral sequences that have been found in the genomes of like chimpanzees, and as Zimmer puts it, quote, since our ancestors split from theirs about 7 million years ago, this shared viral DNA must come from our common ancestor with chimpanzees. And Zimmer goes on to describe research that has been able to trace the invasion of our genome by viruses to about 30 to 35 invasions, as he puts it. And there are periods, interestingly, during which there seem to be particularly frequent genome insertions, but then also periods over the course of our evolution with lower frequencies, or it didn't happen as frequently. So the study to which he's referring compares a bunch of related primates: baboons, macaques, gorillas, chimpanzees, bonobos, gibbons, and so on. And the study was conducted by Magiarkinis, the last name. I apologize for probably butchering that last name, but it was published in the journal Retrovirology in 2015, and it's available without a paywall online. Like, you can go read it if you want. And so early on in the evolution of our primitive primate ancestor, about 40 million years ago, the study suggests that viruses tended to contribute quite a bit to our ancestors' genomes. And not only that, but the integration of viral sequences into our ancestors' genomes happened quite a bit more rapidly than they have more recently, right? Those periods of high frequency and then periods of low frequency. And in fact, as I mentioned earlier, humans may never have been able to evolve at all if it were not for these instances of viruses integrating into one of our ancestors' genomes. And so here's an example, and it's pretty cool, of an essential point in our evolution where infection with a virus fundamentally changed the way life could adapt at all to new environments. So one of the features of the biology of mammals that's basically sort of central to defining the class, after all primates are mammals, is the use of a placenta during the growth of offspring. So not all mammals use the placenta, but certainly when it comes to like the primates, certainly when it comes to primates and probably mammals of which you could think, all use placentas. So cats, dogs, rodents, cows, and of course humans all use placentas. And so the placenta was like a gigantic evolutionary development. It's what enables two humans that are like genetically pretty different from each other to contribute genetic material to their offspring without the mother's immune system attacking it due to the presence of some other human's genetic material, right? That's what the immune system does. And so, without getting too far into the nitty-gritty biology here, a pretty critical component of the placenta is composed of cells that fuse together, right? Forming a sheet of tissue. And in particular, we're going to focus on the outermost layer of the placenta. And it has one of those unforgivable biology names that I've actually never said out loud. I've only ever read. Syncytiotrophoblasts, I believe is how you pronounce it. Or maybe it's pronounced syncytiotrophoblastic. I'm not sure. I'm not sure which is more commonly used. And so, okay, anyways, where did viruses come in to this layer of tissue? One of the proteins that enables these cells to come together and fuse to form that layer of the placenta, it doesn't seem like it came from human DNA. Rather, the sequences that compose this protein seem to more closely resemble the sequences of viruses. So this protein that we're talking about is syncytin, or it might be pronounced syncytin. The name doesn't really matter. Don't worry about it. What does matter is that there seems to have been, or the sequences seem to have been derived from some kind of virus. And in fact, it may be derived from one of the mechanisms by which viruses first attach to cells that they then later infect, right? So you might have heard about, if you've been paying neurotically close attention like I have, you might have heard about the spike protein that SARS-CoV-2 uses to infect our cells to cause COVID-19. And so what we're talking about regarding the connection of placentas to viruses is like that spike protein. So how could this have come about? right? One potential way was that several hundred million years ago, some egg-laying vertebrate was infected by some kind of retrovirus, sort of like HIV, again. And rather than doing what viruses typically do, and it made its way into infecting, just this rare instance, this virus made its way to infecting that egg-laying vertebrate's egg cells. So rather than dying from the infection, that egg-laying vertebrate ancestor ended up reproducing. And voila, the offspring of that fateful moment of reproduction produced the, old ancestors of what eventually would become mammals. And so that offspring just, likely just happened to be able to create more effective barriers, placenta-like barriers, as a protective layer over its developing offspring before birth, and was therefore more likely to successfully generate offspring. And so due to that developmental advantage, this virus-derived feature of their genome ended up being evolutionarily favored. And so, interestingly, this is only one of many examples of virus-derived genes being integrated into the genomes of animals, and those ENV sequences from viruses in particular. So ENV, think that, like spike protein from SARS-CoV-2, the thing that it needs to attach to a cell to infect. So, one way to think of it is that our ancestors were able to sort of like domesticate these viral sequences and use them to their reproductive advantage. So, this contribution of viral sequences to our ancestors' genomes happened at a pretty steady clip of the millions and millions of years of the evolution of like old world monkeys, or called old world monkeys, meaning things like macaques, baboons, mandrills, and one of my favorite primates, the proboscis monkey. Never seen that? Check it out. Pretty hilarious. And in fact, humans likely could have never arisen were it not for a viral infection of one of our ancestors millions and millions of years ago. And so, but when it comes to the evolution of apes, so meaning chimpanzees, bonobos, gorillas, gibbons, orangutans, and of course humans, the rate of viral contributions to the evolving genome dropped lower in every successive branch of evolution, meaning that as apes continued to evolve, viruses tended to be integrated into our ancestral genome less and less. So why might that be the case? What's the deal with this change in frequency of viral integration? Why would the rate of viral sequences being integrated into the genomes of monkeys stayed about the same while it continually dropped as apes evolved? So we don't know for sure, but there are some hypotheses. So first, it can help to compare the broad biological differences that distinguish apes from old world monkeys. So one of the more obvious differences is that apes just tend to be bigger. And at the simplest level, that means that there are literally like more cells that compose a given ape than do any given old world monkey. And so with more cells available comes a greater possibility that one of those cells might start misbehaving and becoming carcinogenic. So in other words, it may just be easier for cancer to begin to grow in a larger ape than in a smaller monkey. And as a result, anything that might predispose a cell to begin to misbehave in this way is more likely to be selected out. Or, so if the integration of these novel viral sequences happens to have some possibility of causing that kind of aberrant cellular activity, not 100% of the time, but just occasionally, the presence of more cells presents more opportunities for that possibility to result in the growth of cancer. And so as a result, while a fairly low probability of cancer growth may not have, fatally threatened an animal with comparatively fewer cells, that low probability may have been enough to threaten the survivability of animals with more cells. So as a result, an ape that's been infected with a virus that has, a tendency, even a fairly low tendency, may end up with a lower likelihood of reproducing. And so, even if the ape, infected with this possibly carcinogenic virus ultimately reproduces, their offspring would also be afflicted with this increased probability of developing cancer or other maladies. So therefore, reducing the likelihood that those genes that have viral sequences integrated have just a lower likelihood of being passed down than the genes of other members of the species who've not been exposed to that same virus. And so similarly, it may be the case that this greater number of cells has tended to select for the development of even better defenses against any kind of viral invader. So, while the prospect of viruses being integrated into the genomes of smaller primates, like, old world monkeys, doesn't necessarily imperil their ability to reproduce. It may be more likely to imperil the abilities of larger apes to reproduce. So given the viruses are basically everywhere, it may have basically been more likely that larger apes would develop more sophisticated immune system defenses against a viral intrusion. So, in other words, it just may be the case that the bigger you are, the more important immune systems become to defend against viral intruders and or aberrant cellular activity that arise from those viral intruders. Another possible explanation, though, may arise out of the evolution of our brain. So, which is where my discipline comes in. But more specifically, the evolution of our capability to use tools and social cooperation to get food, interact with our environments, and fight off other animals and other humans. So viruses spread by a variety of mechanisms, through sexual contact, respiration, as evidenced by the global pandemic we're currently confronting, and importantly, through the blood. So let's use HIV as an example. It's pretty ideal in this respect. As I mentioned earlier, HIV is a retrovirus capable of integrating into the host genome of the cells it infects. So a virus that's sort of similar to the types we're discussing, right? A retrovirus, the type that can integrate, right? So generally speaking, HIV was spread sexually. Though it can also spread through exposure to bodily fluids in general, including the exposure to blood that contains the virus. And so this is why, in addition to being able to spread as a sexually transmitted infection, it can also spread through the sharing of needles for intravenous drug use. And so, while we don't really rely on our teeth as much these days for self-defense, our evolutionary ancestors did rely on their teeth in a fight for survival. That's one of the best tools they had. And so as a result, if a primate was engaged in some kind of battle, either for food or for defense, and chomped down on their opponent, successfully biting into them, they'd be directly exposed to a whole bunch of the other animal's blood. So, however, as our ancestral humans developed the ability to use tools, either by gathering plant-based food or using tools to hunt, particularly in groups, right? That super efficient mechanism by which they'd be exposed to another animal's blood became dramatically minimized, right? They just didn't use their teeth, their mouths as much. And so even in fights with other humans, using weapons of some sort, even crude weapons, mitigated exposure to another human's blood. So this is evidenced by the size of our canine teeth relative to other primates. So, we just don't need to have fangs as much as, the primates that hadn't developed this versatility through the use of tools or weapons, through cognitive evolution. And so we have smaller canine teeth relative to our primate cousins. And this is true even if you look at the canine teeth of chimpanzees and bonobos, our closest primate relatives. They have some pretty ferocious canines, but all throughout, the great apes. Check out, I should have included some pictures. I'm kind of kicking myself for not doing it because they're pretty ferocious fangs, way bigger than humans. So, as a result, the emergence and expansion of cognitive capabilities, enabling us to use tools and cooperate with one another, may have significantly minimized one of the primary ways by which these viruses that can integrate into our genomes are able to be transmitted. Our ancestors just weren't being exposed to as much blood. So, we can't know for sure if our relationship with viruses has come largely to a close, if that frequency of virus integration among the whole gene pool has dipped to basically zero. Or if, much like COVID-19, a novel retrovirus will emerge in some kind of reservoir species that's able to exploit one of the fundamental ways by which modern humans, eat, reproduce, or live our daily lives. It's tough to know, but I think it's just an interesting chapter in the story of human evolution. A pretty non-trivial part of your genome. Again, somewhere around 8% of your genome, the genes that are being transcribed, translated, and expressed as we sit here chatting, is derived from viruses that snuck into the genomes of our ancestors and now have become a part of our genetic legacy. Okay. That's what I had for you today. And I had to stop myself before going into the theories of the origins of viruses, which is another super interesting story. I remember one of the first science-y books I ever read, and this was, I guess I was in junior high school, was a pretty short, pretty concise exploration of various theories regarding the origins of viruses and how they pertain, how they relate, how they interacted with the origins of cellular life. I also would have loved to have gotten into the various arguments about whether viruses should be considered alive and another branch of the tree of life, or if they should be, conceived more of like as being like a vine that's climbed all over the tree of life rather than formed its own sort of branch. And so, maybe next time we'll get into that. But if you've stuck with me this far, I'd appreciate it if you'd subscribe, for more discussions like this. I do this at least once a week, almost always on Sundays. And thanks for watching. And now we will start the live half. All right. Hello, everyone. My name is Ian McLaughlin, and I am a PhD in neuroscience from the University of Pennsylvania. And on YouTube, I just talked about your viral legacy, the nearly 8% of your genome that's derived from viruses over the millions and millions of years of our evolution. And it's a really cool story. I also talked about some viruses that are so unique and so gigantic that they begin to blur the line between what's considered life and what's considered not life. So you can find my YouTube channel by going to, searching like Ian Anthropoid Brain, or it is youtube.com/anthropoid. And if you, like I'm sure many people don't really find the spelling of anthropoid intuitive, you can find that's my handle on Twitter and Periscope is anthropoid. Oh, yes. I don't think I will ever remember to do this. It's just easier for me to see your comments if I do this. Here we go. And do that. Okay. So how's everybody doing? Hello. Let's see. Asha, good to see you, Asha Valani. Hello and good evening to you. I hope you're doing well. Hi, yeah. Hey, Asha. Okay, there we go. Likewise, thanks for coming by. Yeah, so it's a really cool story, and I also talked a bit about, again, this is on YouTube, I talked a bit about a sort of an essential component of our evolution without which humans never could have even come into existence, let alone like mammals, and derived from a virus. And we have a pretty good idea of what the virus probably looked like. So yeah, it's a cool story. It's one of my favorite I've done so far. Can I ask about memories? Of course, you can ask whatever comes to mind. I'll do my best to try and provide some kind of satisfying conversation, but certainly. Long time since I've been on live. Hope you're all well. Sinead from Northern Ireland. Great to see you, Sinead. I hope you are doing well. How'd the thesis go? Great. It is donezo. The dissertation is finished. I guess I never actually showed my dissertation book on live. But anyways, it's in a different room. But yeah, it's good. It's way too long. It's like 400 pages. But more recently, I just wrapped up probably what's going to be my last like scientific publication from the lab in which I've been working. Okay, when I was young, I was exposed to lead and mercury. This is a question from YouTube. As a safety engineer, I inspected factories, breeding ground for many industrial solvents. Could this contribute to face blindness? Okay, we need more paleo epidemiology. Yeah. So a lot of what I talked about on YouTube is considered like paleovirology, and it's basically like a new branch of paleontology, because obviously they're studying viruses, which are not sort of the typical things that paleontologists study. Thank you so much for the super hearts, Joy. I really appreciate it. So face blindness. Prosopagnosia, I believe, is the term for that. Can exposure to heavy metals bring that about? So, I mean, theoretically, yes. Basically, the reason that exposure to heavy metals is so dangerous is, we use a bunch of different atoms and molecules in our brain, right? Perhaps most importantly, are ions, sodium ions, potassium ions, calcium, in our neurons. That they're like they're like the words that neurons use to communicate with one another, rather than the sentences. No, they're more like the sounds that neurons use to make words, right? Anyways, and those ions literally have to flow in and out of neurons very rapidly. And so when you have one of the ways that exposure to heavy metals like lead or mercury disrupts neuronal function is that they can basically disrupt the ability for those other ions to flow, right? So they can sort of block the sort of channels through which sodium and potassium flow, and that happens, and that's bad. However, another problem with heavy metal exposure is that our neurons don't really have any mechanism by which they can clear out those heavy metals. They just accumulate and sit there. And so that's a problem, right? Now, with regards to its ability to develop this specific malady, prosopagnosia, or face blindness, I'm almost certain that's what the technical term for it is, it's possible. So the issue, you're referring to a syndrome that arises, generally speaking, as far as we're aware, from disruption of a particular part of the brain called the fusiform gyrus. And so this is probably most classically associated with stroke. Some people who have a stroke in that region develop face blindness. And what face blindness means, by the way, for people who aren't familiar, it probably sounds kind of crazy. Like, people with face blindness can see other faces, but they just don't register it as, most other folks with, normally functioning fusiform gyri do. And facial perception for humans is absolutely critical to communication. We devote a ton of our sort of cognitive horsepower and real estate to not only vision, but also deciphering the emotional states, the dispositions of others by evaluating their face, and then also more specifically their eyes. And there's some really wild research showing that some people, or that humans in general, some random sampling of humans, can generally speaking predict of which party a given political candidate or with which party a given political candidate affiliates by just looking at black and white images of their face. And even if they narrow down to just pictures of this, like their eyebrows and eyes, even with just that information, better than chance, humans can predict with which party a given political candidate affiliates, and better than chance, be able to predict which candidate will win in an election before the election happens. Pretty crazy, huh? So when it comes to face blindness, that capacity to sort of decipher and really understand the faces of others is damaged. And so all of this to say, I can't tell you for sure that lead exposure or heavy metal exposure would cause something so specific in the absence of other disruptions of cognition and consciousness, but it is theoretically possible. Okay, you wrote me about the issue. Thank you for the explanation, Xenia. Much appreciated. My pleasure. Thank you. All right. I think I could pass the quiz on this lecture. Awesome. How is there IQ? I've actually never taken an IQ test. I just never had a reason to. And IQ has been coming up fairly regularly these days. There must be some reason. Usually that means somebody prominent has talked about IQ. The IQ test is and using it to compute or determine somebody's intelligence is a pretty contentious topic these days. Generally speaking, from what I can tell by sort of my interactions with other people in the field and observing conversations between other scientists, neuroscientists, it seems to me to be the case that most neuroscientists, and it's not only neuroscientists that study intelligence, but I would submit to you that if you want to understand intelligence, you probably need to understand the brain. But most neuroscientists would argue that intelligence is far more versatile and diverse than the things that the IQ test measures. So in other words, the IQ test is not an optimal gauge of human intelligence. It does, I think everybody would agree, it does measure something very important, some of the most important components of human intelligence. But I think it has this sort of social cachet that is almost certainly undeserved. And partly because the marketing of IQ, intelligence quotient, was pretty brilliant at the time. And keep in mind that the IQ test wasn't even devised initially to be used in the way that it's used today. Anyways, that's why I haven't taken one. But I would be 99.999% confident that my IQ would be right in the middle. Not particularly high, probably not particularly low, right in the middle. So just as smart as you guys, no smarter, and hopefully at least as smart. How do you feel when you come across dumb people? I think I understand where you're coming from with that question. Because I studied the brain, I must be very, very intelligent, and that's just not the case. Science is just like any other discipline. in that it's composed of people who are just your average folks. And they what perhaps distinguishes them is that they have to have some pretty intense enthusiasm and passion about a very specific topic within science, because it is just a crazy amount of work. Like when I was working on my PhD, particularly right in the middle of the of the mire of my PhD, I would regularly spend 14, 16-hour days and come home at like 5 a.m. and not even have my experiments work. Right. So you have to be able to sustain a very high level of enthusiasm. But that is it. I mean, it's not like neuroscientists are genius. Of course, there are geniuses who are scientists, of whom I am not one. but so as a result, I really don't approach other people, new people, with that thought in my mind as like whether or not this is a smart person. I pretty much always assume pretty much everybody's largely equivalent. Some people have tremendous gifts, including cognitive gifts, but by and large, I just assume if somebody has different beliefs than I have, or if somebody misunderstands a scientific concept that I understand because it's my job to understand, I don't equate that with being dumb or stupid. I equate that with just different life experiences, it's always the most interesting conversations for me are conversations on topics about which I disagree with my conversation partner. Okay. Do our brains have anything to do with our sexual orientation? Absolutely. That's a great topic. So any human behavior that you can probably name has to do with the brain. And the principal governing, structure of any behavior you can think of is encoded largely within the brain. Sexual attraction is included. However, sexual attraction is not as simple a facet of human consciousness as I think it is widely appreciated, just because I think it's one of those things that's very sort of, it is, principally subconscious. And it's sort of like intuitive, and you don't really have to like actively think about it very much. It just sort of happens, right? It's a part of the biggest part of our consciousness that's subconscious. So yes. So, and the point being, as a result, pretty much anything that humans do is more complicated than just like, there's no like sexual attraction part of the brain. That's just not how it works. It is the byproduct of like a whole constellation of different brain regions that encode things like memory, obviously libido, and then also, more sort of forebrain, is this a good idea? Is this a bad idea? Risk reward sort of weighing. And socialization, right? Like, what the environment into which you were born, the society into which you were born has sort of indicated to you is normal sexual attraction. But all of that does happen in the brain. It's just not so simple as like one specific structure. And if you're interested in sexual attraction and in love, I have a podcast called Wired to be Weird, and I have a podcast episode about love. Why do we fall in love? What is it about the, neurobiology of love that enables us to engage in all of these like objectively totally irrational behaviors just in pursuit of love or in maintenance of love. And there's some really interesting research that I get into, regarding the role of dopamine in distinguishing why it is that some people stay in marriages very happily, or at least part of the reason that people are able to stay married for a very long time and sustain love, whereas other people don't. What up, big homie? What up, man? Neck face. What is the most fascinating, groundbreaking research taking place in your field right now? Well, so, we are fortunate to live in what some have coined a golden age of neuroscience. And so there's just a ton of really cutting edge, stories that will be coming out every year moving forward for a while. And that's because there's been this dramatic proliferation of tools that we can use to ask questions that would have seemed like sorcery 10 years ago, 15 years ago. Definitely like what I did in my PhD absolutely would have struck me as being sorcery when I was in college. Being able to like control very specific neurons essentially remotely in a living, waking organism. I mean, it just seems like sorcery, but it's totally possible. In fact, it's almost mundane at this point. So for me, I think that is some of the most critical research that's going on right now, including or continuing to expand the number of resources available to a neuroscientist. But, and I'll just give you some examples. So the integration of organoids into the research of neurobiological, diseases and disorders that are based in the brain will almost certainly bring us significantly closer to understanding the basic biology of things like schizophrenia, bipolar disorder, these types of mental illnesses, than we could have otherwise. And what is an organoid? An organoid is basically like if you take, so in neuroscience you have some options. If you want to study, let's say, schizophrenia, you can have a model of, you can study humans with schizophrenia, right? Do some neuroimaging, evaluate their behavior, these kinds of things. Or you can study an animal model of schizophrenia, right? Where animals, tend to exhibit a lot of the same things that humans with schizophrenia exhibit. Or you can study essentially neurons that are derived from somebody with schizophrenia in a dish outside of the brain, outside of any biological system. And, they're all very, very valid and reveal very important features of something like schizophrenia, but they all have advantages and disadvantages. And there is this gap between studying any biological dynamic like schizophrenia in a biological system versus in a petri dish. And what organoids do is basically, if you were to take those cells that would have otherwise been in a petri dish and you implant them into a biological system like a brain, those neurons will behave far more similarly to how they would behave in a normal, the brain of somebody with schizophrenia. So there's that. Organoids, very important. CRISPR-Cas9, also going to be, already has been, extremely important in neuroscience research. CRISPR-Cas9 is a technology that enables us to very precisely, very efficiently, very accurately modify genomes, modify genes. And prior to CRISPR-Cas9, we could still do that, but it was far less precise. And so now, I worked with animal models that were produced using CRISPR-Cas9, but inevitably that technology is going to yield a huge number of novel treatment strategies for conditions that are based in genetics. And after all, a lot of conditions are based in genetics, it's just the genetics are extremely complicated. Okay, there's that. There's, whole brain-computer, or let's just say brain-computer interfaces. The integration of smaller and smaller and more precise mechanisms by which a computer, essentially, can receive information directly from the brain, from the entire brain. Those technologies have been in development for years and years, but they're beginning to get to the point where they're sophisticated enough that we're getting closer to something like Neuralink, right? Something where you can buy a thing that you implant in your brain, and then all of a sudden the bandwidth of your ability to communicate with the computer is dramatically expanded. And I talked about that last week a bit. And there are so many others, but there are just too many to name. I think objectively, given the past, we must be in something like a golden age of neuroscience, although I suppose you can't technically identify a golden age without being able to look back in retrospect, But I think I would imagine that, we're in something like that. He's a pseudo-intellectual and is not as smart as you think he is. Well, I am not particularly smart. I'm no smarter than you. I'm adamant and confident that that's the case. And I guess it's not for me to decide if I'm a pseudo-intellectual. I don't really consider myself unintellectual. I'm just a scientist. All right. Thank you, sir. Super interesting. That's my pleasure. Thanks for coming by. Okay. Why can't our brains keep the important information and skills we studied in university? Agilma. Awesome topic. Yeah. The number of things that I have forgotten since starting college and graduate school is so much greater than the number of things than I actually remember. Okay, so why is that? So essentially what we're talking about is human memory. Human memory is magnificent. It is an absolute, one of the pinnacles of evolution. Essentially, it gives us the cognitive power to cognitively travel back in time. That's pretty wild. You can't, not your entire body going back in time, but you can bring yourself back to the past. So that's magnificent, and human memory is a lot better than a lot of other animals. However, it is fallible, right? It is not perfect. Human memory is notoriously error-prone, and in part it's because part of the systems that encode our memory not only operate to preserve experiences, but also to forget experiences that are not deemed to be essential, right? So it's important to be able to forget, in other words. And there are various theories as to why that happens. But fundamentally, you might have heard of neuroplasticity, right? So the part of neuroplasticity with which I think most people are familiar is synaptogenesis, is the creation of new connections between neurons that didn't exist before, or the strengthening of connections between neurons. So that's one side of the coin of neuroplasticity. The other side is the loss of those synaptic connections via a mechanism called long-term depression. So long-term potentiation is critical to the formation of new synapses, whereas long-term depression drives the pruning away of old synapses. And by and large, at a simplest level, the things that determine whether or not a synapse is going to undergo long-term potentiation or long-term depression is how active it is. It's in other words, it's like use it or lose it. And so as a result, we learn a ton in university, right? It's a tremendous privilege and a luxury that human society has afforded us this capability to learn, to spend time focusing on learning. That's fantastic. But if you don't touch those synapses periodically, just inevitably and automatically, those synapses will begin to undergo long-term depression, and ultimately they'll they'll not necessarily be wiped from your brain, but just be more and more difficult to access, right? Because it's just those synapses are not particularly strong. So this is a very reductionist explanation of why this happens, but that's partly why it happens. So you just have to try and just go back to those things that you want to remember and just brush those cobwebs off those synapses and revisit that information. I know my wife has saved all of her notes. She went to a fabulous college, and she's brilliant, and she saved all of her notes, and it's wild. I mean, it is. Let me tell you, it's a pain when you're moving, but it's just this wonderful exercise to go back in time and revisit all that information. Great topic. How to spell synapses? So yeah, like that. you spelled it correctly. How do you explain depression? Great topic. So up front, we don't actually know exactly. Thank you very much, Brian. I appreciate the super hearts, man. And thank you for that, for reminding me. So we don't thoroughly understand what it is about the activity in the brain of somebody with depression that distinguishes it from the activity in the brain of somebody without. We have some ideas, though. And our understanding of depression has evolved tremendously over the years. I mean, we went from not really having a concept of it even existing to essentially, calling it different things. And then we came to ultimately what was called the serotonin hypothesis of depression, and that arose because it was observed that certain antihistamines had what seemed to be an antidepressant effect. And you might be familiar with diphenhydramine or Benadryl. Think that. And it turned out that in at least some people, their symptoms of depression were attenuated by this antihistamine. And so, back in the day, medicinal chemists went through all the known pharmacological activity of these drugs, and then essentially honed in on developing drugs that would be able to target serotonin in particular, and thus fluoxetine was born, one of the best-selling drugs of all time. Fluoxetine is Prozac. And it is moderately effective, but I can get into the theories of why we think Prozac does what it does. But moving forward several years, we have sort of come to appreciate that depression is far more complicated than any one neurotransmitter. It's almost inevitably going to be a byproduct of the activities of specific circuits, probably in the ventral midbrain, some of which release serotonin, but some of which release dopamine, norepinephrine, all of the monoamine neurotransmitters, all the neurotransmitters of which with which you're familiar, like GABA, glutamate, etc. And it's not the neurochemistry, it's not the neurotransmitter levels that's the most pivotal factor. It's the activities in these circuits, and those neurotransmitters are just one character in the story. And so I happen to have studied in my thesis, my PhD research. I happen to. Whoa! Oh, sorry. Siren. Anyways, one of the circuits that we believe is sort of pivotal in encoding depression like activity. Okay. What are synapses? So synapses are okay. Think of a neuron as like, well, it's sort of like an amoeba, I guess, but just a round belly, right? An orb. And one of the things that distinguishes neurons, the principal brain cell with which we're concerned when it comes to the brain, not only brain cell, but the main one, neurons, what makes them so cool is that they can communicate with other neurons. This neuron becomes active, and that can cause this neuron to become active or to silence. And the way that happens, principally, is through these projections, these connections. Think like, cables or the charging cord for your smartphone. But instead of directly contacting another neuron, although that does happen sometimes, there's a tiny little space between the points at which they talk. So it's sort of like if you were holding your charging cord just outside of your smartphone. And it is at those points of communication where this neuron is talking to this neuron that neurotransmitters are released, and some of those ions that I was talking about earlier are in flux. And it's that point of communication, that little space. Well, that point of communication is called a synapse. The little space is called the synaptic cleft, but who cares? Hey, BCM nurse, good to see you. I assure you I'm not a genius, I promise. Simply Amanda, well, thank you. I'm happy to see you too. What are the effects of 5G and the brain? Okay, yeah, so the short response is that there's no evidence that there's any effect of 5G on the brain. There's no actual concrete scientific evidence, right? And I sort of observed this hypothesis that 5G, upon its inception, will damage human biology and cause some kind of disease and pathology, and then it was associated with COVID-19 or the novel coronavirus. And so here's the issue with that whole hypothesis. What 5G is, it's just a point on the electromagnetic spectrum, right? You might be familiar with microwaves, gamma rays, Wi-Fi, cellular signals in general, Bluetooth. All of those are different points on the electromagnetic spectrum. And probably the point on the EM spectrum with which you're most familiar is visible light. That's also on the electromagnetic spectrum. Visible light doesn't damage the brain. Visible light doesn't damage the brain or damage your body. And the only reason we're even aware that it exists is that we have very special proteins in our eyes that are sensitive to that form of radiation. So I'm very happy that we evolved that capability, because photons, which are what's delivering all this light, they travel very, very quickly. In fact, at the speed of light. And so it's a very up-to-date update on what's going on around you, because photons are constantly crashing all over us, and we can use these photoreceptors, these specialized proteins, to determine the spatial organization of those photons, and thus we can see. But that's the only reason we know that visible light even exists, is because we have those proteins that are specifically sensitive to that point on the EM spectrum. If those proteins were sensitive to, X-rays, we would see in X-rays, which I think would be very cool. Like, once Neuralink exists and we can integrate some computers into our brain, I'm definitely going to get the X-ray or the gamma ray feature, where part of your sensory experience can be like influenced by so then you could like see through walls potentially or whatever. So we have these specialized proteins. Now, some parts on the EM spectrum do harmfully interact with biological systems, in some cases causing cancer. However, the vast majority, for any part on the EM spectrum to meaningfully interact with a biological system, the physical properties of those waves of radiation have to be such that they can interact spatially with some some cell or something like that. And there's just no evidence that 5G, 4G, 3G, any of them meaningfully interact with biological systems. Now, as a scientist, I don't approach these questions by saying, oh, that's ridiculous, because it didn't come from, an Ivy League scientist. It didn't come from, some, scientist at all. Like, I never totally discount any hypothesis, but just like everything in science, just like my colleagues expect me to, when I make a claim, a scientific claim, you have to bring evidence. And evidence is gathered by conducting experiments, by coming up with a hypothesis, testing that hypothesis, coming to a conclusion about the hypothesis. And I've seen zero evidence of that in any of the conversations about 5G that I've seen. I've seen physicians talking about 5G in ways that they're just connecting dots. Like, they have no evidence. More evidence that physicians are not scientists, just different kind of training. So, hey, I will totally change my perspective if somebody provides some evidence that 5G meaningfully interacts with the brain or with biological systems. But until then, I think of it the same way as if I were to tell you that, this paper is made out of gold and it's worth $50 million, and I'll send it to you for just $10 million. Like, prove that. Okay. Hello, Yuka. How are you? Are there any researchers that are particularly inspire you at the moment? Yeah, for sure. So, there's a bunch. Okay. So in terms of like researchers that most directly inspired my own personal research, Brian Roth at UNC is a wizard. University of North Carolina. He invented a bunch of different technologies, biomolecular technologies, most notably for me, what are called DREADs, or designer receptors exclusively activated by designer drugs. It enables you to just remote control neurons using pharmacology. But he's come up with a bunch of different things. So his whole labs is very inspiring to me. Then, of course, there's Carl Dieseroth over at Stanford, I think. He invented optogenetics, or he didn't invent it alone, in fact, but he was one of the people that invented optogenetics. And then his collaborator at the time, Ed Boyden at MIT, both of their labs are just incredible. They're both like Willy Wonka's, chocolate factory. Like, every year they come out with just these mind-blowingly impressive technologies that enable us to, get more and more granular about the relationship between brain activity and consciousness, right? After all, that's what I'm interested in. Who else? Jennifer Doudna, the person who's credited with most directly inventing CRISPR-Cas9. She's also brilliant. Beyond just, sometimes scientists come up with this wonderful idea, but at least a big part of it was they're in the right place at the right time, right? But for her, she's just brilliant. There's Jen Heemstra. Oh gosh, where is she? She's at, oh man, is it like Yale? No, I can't remember. Jen Heemstra. So she's a great neuroscience researcher, but beyond just her elegant science, she's also, I think, a very, very good mentor. I think there are very few in science. There's kind of a paucity of very effective mentorship. Nobody teaches you like management when you're training to be a scientist. But that doesn't mean that fabulous mentors are absent from science. There are some, and my mentor was also absolutely fantastic. But and so is Jen Heemstra, and just she like really emphasizes the importance of teaching and mentorship, and I think that's very valuable, not just to the scientific community but to society more broadly. Okay, who else? I'm forgetting so many people. Oliver Sacks, not technically a neuroscientist, but I think the late Oliver Sacks. I think he had a very unique approach to communicating and understanding diseases and disorders of the brain, and he has written some of the most important books in the history of neuroscience. There's a movie about his work called, I think, Awakening. So it's a great movie. It's really cool. He's a really creative guy. God, I could go on and on. There are just a ton. Gosh, who else? Elliot Tucker Draub. I bring up his research all the time, particularly when it comes to when people are interested in the relationship between genetics and IQ. I think he's done some of the most comprehensive work towards getting us to a more accurate answer to those questions. I really like Joseph L. Graves. So he's at University of North Carolina. I can't remember which specific campus, but UNC is a fantastic set of institutions, and he, I think, is literally the best scientist in describing and understanding the relationship between genetics and what we call race. I think he's just the best. I could go on and on, but there's a bunch. I mean, we are fortunate to live in an era where there are a lot of really brilliant scientists, but the problem is that we're also living in an era where, at least in the United States, the government has decided to stop investing. I want to be precise when I say that. The government is investing less and less in biomedical science and in sciences more broadly. The numbers seem to go up, right? But because of inflation, the purchasing power of the dollar, relative to where we were 5 years ago, the investment is lower, right? Because you just need more funding to acquire lab equipment, to pay people. And also, studies out of Stanford have shown that as a science becomes more and more sophisticated, the level of investment necessary to make the next discovery is going to be progressively greater. So, the more we understand about the brain, the more expensive it's going to be to understand more about the brain. So yeah. I usually go under Ken Norton. I'm so lucky to have this issue clarified. As an artist 20 years old has the same issue. Oh, interesting. Okay. So of course I didn't realize that was you. Good to see you, man. Oh wait, where is that? Thank you. So many great minds. Much to be hoped for. Yeah, absolutely. If an AVM is cauterized, can it regrow? I'm not 100% sure what you mean by AVM. Sorry about that. What's the best cure for ADHD and or the best ways to manage it, natural and or prescribed? 8, and then is it 8 8s? Because if it is, I would like that. So ADHD is, used to be called ADD, and it has sort of bedeviled psychiatrists and neuroscientists for a while because there is a particular class of drugs that is very good at treating ADHD. And there's been this consistent interest in finding more options because, fortunately, for better or for worse, that family of drugs are psychomotor stimulants. They're dopaminergic psychomotor stimulants, and most specifically amphetamine, which is Adderall, or methylphenidate, which is Ritalin. And then there are some other sort of, slight modifications, like there's Vyvanse, which is like a molecular modification of the amphetamine molecule that makes it a sort of a slower onset and termination. There's Concerta, which is really just like a physical pill structure to deliver methylphenidate more effectively. But by and large, those are the two. And it's not for a lack of effort, right? I mean, definitely groups have tried to evaluate, selective norepinephrine reuptake inhibitors, Strattera, just other pharmacological strategies, and it's never been shown. Or, Provigil, what is it, modafinil, other drugs that do have some stimulating properties, but have slightly different pharmacologies. And just almost without exception, no other drug has been shown to be nearly as effective as these other medications. Now, that might be because it just so happens that ADHD arises from a specific pattern of activity in the dopaminergic circuit in the ventral midbrain called the mesocorticolimbic dopaminergic circuit, and guess which structures in the brain those two drugs target? That circuit. So it might just be like, hey, that's a pretty ideal treatment. So and then on top of that, you brought up like more natural remedies or let's say psychotherapeutic approaches. What has been shown is that so psychotherapeutic approaches alone are not as effective as the pharmacological approaches. And keep in mind, we're talking about like, physician diagnosed ADHD, right? One or multiple physicians have determined that this person has ADHD. So yeah, it's been shown that like behavioral interventions, these types of things aren't as effective. However, when combined, when you have some sort of psychotherapeutic approach, some sort of behavioral approach, coupled with pharmacological strategies, the dose necessary of the pharmacological strategies, again, Adderall or Ritalin, can be lower and still be as effective. So there's that. So yeah. So basically, it doesn't have to be an either or. It can be a both and. And in fact, some of the best results will be derived from a both and, from both behavioral, psychotherapeutic approaches and a pharmacological approach. And if you're also interested in like supplements or things like that, there's a bunch of reasons to be skeptical of the supplement industry, and I can go into them if you'd like. I'm more than happy to. But the short answer is that due to the lower threshold of sellability, almost the lower threshold beyond that somebody has to pass to prove that their product does what they say it does. There's just never been a supplement that has been identified, through like scrupulous scientific research that's as effective. So yeah, in short, coupling psychotherapeutic, behavioral interventions with pharmacological interventions. Hey, Clowny Cans, good to see you. Any successful pharmacology for Asperger's? So when you say like successful pharmacology, and then we can talk about nootropics if you'd like. It depends on what your goal is for Asperger's, right? Because not everybody's goal is the same, and I think it's worth acknowledging that. Not everybody wants to be cured of Asperger's syndrome or, autism spectrum disorder. In some cases, however, the severity of, that can be, sufficiently debilitating where people do become interested in a cure. But I, from my personal sort of philosophical perspective as a scientist, my goal would not be to just totally, cure someone of Asperger's. Rather, it would be to help them to mitigate the more the aspects of their unique brain structures that make operating in sort of many social situations prohibitively difficult, right? So just to improve their quality of life, and so when it comes to that, it's kind of impossible to say like this is the perfect medication to achieve that goal. There are Asperger's, much like autism spectrum disorders. Some of the symptoms are things like psychiatric conditions like anxiety, severe anxiety, obsessive compulsive disorder, these types of things, and there are some effective treatments for those. But it's not like there's a special medication for either Asperger's or autism spectrum disorder. Now we can talk about like some of the more kind of basic research in understanding what it is that's unique about the genetics of somebody with ASD or something like that. But yeah, it's so. I hope I meandered to something reasonably coherent there. Hey Ian, questions. This is on YouTube, which you can find youtube.com/anthropoid. I talked about this really cool, essentially the 8% of your genome that is derived from viruses. Pretty cool, huh? So many moons ago, you were going to quit beer, alcohol, status, music, neurological effects, implication of why someone chooses A or B, your listening habits if you care to share, sure. So a while ago, I talked about a study, and actually I mentioned this last week, I believe, talked about a study out of England that showed that essentially no amount of alcohol consumption is benign. So for a long time in biomedical science, it was kind of presumed, frankly, that heavy alcohol use and maybe also moderate alcohol use, on balance, is deleterious to health. But for the longest time, it was presumed that mild alcohol consumption is actually either benign or maybe even good, like maybe even has some benefits in reducing stress and this kind of stuff. But this study, which was the biggest of its kind ever, basically showed that it doesn't matter, like no amount is without some cost. Actually, it's funny enough that you bring this up. There was a study that came out, and this always happens, right? In these kinds of questions, aspects of our daily lives that are, like so common in human society. Same thing happens with like caffeine, for example, or coffee. Like 1 year it's bad for you, 1 year it's good for you. And so there's a study that came out, I think, like last week that showed that mild alcohol ingestion is benign. So, you should never like completely radically change your life from just one study. But after that study came out, I definitely did reduce my alcohol intake very substantially. And so as a result, I have this store of really fabulous beer. Like I have some Belgian Trappist ales that are now probably on top of like the 5 years of aging that they do at the monasteries, are now probably an additional 5 years old. So, I told myself I was going to drink one of them when I finished my PhD. Never got around to it. No more guzzling Chimay. No, and I do have some Chimay in there too. And I have some Heady Topper. If real beer nerds will know what that is. I have a bunch of Heady Topper actually, and Focal Banger. So these are beers that are from The Alchemist, a sort of legendary, one of the best breweries in the world now for years and years at this point in Vermont. I also have another one that I'm forgetting right now. But anyways, yeah, I have all them. I haven't touched them for a long time. But every now and then, I'll have like, a sort of less rare beer. And then okay, for music, neurological effects implication, why somebody chooses A or B, right? Why somebody likes this style rather than the other. Gosh, I really need to review these notes, but I did a podcast episode on that. Like, first of all, is there sort of like a language to music? Meaning, are there consistent features of music, regardless of genre, that are consistently conveying the same interpretation to the listener? And for a very long time, it was thought yes, and that it doesn't matter what genre it is, a certain progression of notes always sounds sad. So like minor chords, for example, were presumed to always sound kind of sad. But that turned out to not be the case, and it was very dependent upon the culture from which somebody was listening. And then very recently now, I guess not that recently, I guess it was probably like 3 years ago, I did a podcast episode on a study that came out that basically showed through the use of like big surveys of people and in a far more sensitive index of music, that showed that yes, there are some pretty consistent features to music that are cross-cultural. Like, a fast driving beat versus a slow beat versus, these types of things. You can check it out. It's on the podcast, Wired to be Weird, on iTunes and Stitcher. Anyway, so, and I think they do get into a little bit of like why somebody tends to gravitate towards some styles over others. But by and large, I think the vast majority of neuroscientists would suggest that it's almost entirely about your socialization. Like, when you were growing up, what the most popular songs were, what your friends listened to, the experiences with which you associate different genres of music. Like you had your first kiss when you were listening to Kiss or something, And so as a result, whenever you hear Kiss, you think about that sort of exciting period of life, and otherwise, right? And then my personal music listening habits, I'm all over the place. And I've tried to impart that kind of diverse taste in music to my daughter. So, she's listened to Jimi Hendrix, John Coltrane, Miles Davis, Led Zeppelin, who else? Metallica, but then also like Bonobo, and Daedalus or Borders of Canada, these types of bands, Caribou. So it's just all over the place. Lately I've really liked Nine Inch Nails, which I'm sure like somebody who hasn't listened to Nine Inch Nails for a very long time probably thinks, oh wow, geez. But I think Trent Reznor is like legitimately a genius, and him working with Atticus Ross I think is awesome, and they're just fabulous. I really like their music. I like all of his music. So that's what I've been listening to most recently. As I was finishing up my the last paper, that's what I was listening to. Thanks, check it out. Have listened to your episode before. Right on. Yeah. It's a cool study. It's also kind of a simple study that they conducted, but hey, it was well conducted, or well devised. It's a cool question, right? I think everybody wonders that every now and then. Hey, what's up, Daryl? Good to see you. But it's fascinating that one type of music resonates with some, but not with others. Yeah, exactly. And I'm struggling to remember if they did come to any conclusions about like, personality types that tend to aggregate with certain styles, cross-culturally, like, fast driving beats, very, melodic singing or melodic singing, or chorus style singing, or, aggressive, simple singing. I think they did. And there were like personality predispositions that then could, segregate. And maybe I was just projecting that on, because that's what I would imagine. But marijuana, I think it does not affect your brain. So Gustav, the only reason anybody would really ever consume marijuana is because it does affect your brain, for sure. Like, the reason you get high is because the state of your brain has changed. That's not intrinsically a bad thing. I think the question that concerns most people is, do those changes over time, that altered state of consciousness over time, does it translate to permanent changes in like the structures of the brain? And relative, and I think it's always really important when you're talking about cannabis, it's really important to acknowledge that there are other drugs that people do recreationally that society at large has decided are totally legal, and it's up to you as an adult to make the decision as to whether or not you want to consume that drug. Like alcohol, we were just talking about. Alcohol definitely damages the brain permanently. The degree to which that damage is significant varies. And of course, it's a byproduct of amount consumed, people's genetics, stuff like that. So alcohol does, and it's legal throughout much of the world, right? What else? Whatever. And so cannabis, there's no evidence that cannabis damages the brain in that way, in destroying neurons. That doesn't mean it's benign, because there is some evidence that suggests that over time, again, we're talking about years of use, that like broad anatomical changes, the trajectory of neuroplasticity, right, of how the brain evolves over time as a function of experience, is modified by chronic cannabis use. Specifically, like, changes, and I brought this up last week, changes in the densities of white matter to gray matter. And that's a meaningful change. I mean, it's the degree to which it is a permanent change is still contentious. But I think, throughout the entire scientific community, neuroscientific community, including the director of NIDA, Laura Valco, we're all on the same page. Like, it comes down to this: if alcohol is going to be legal, cannabis should be legal. There's no justification at all that cannabis is not only illegal but Schedule 1, the most illegal that a drug can be, whereas alcohol is totally legal. Doesn't mean, but once again, and like, and think of it this way: like, alcohol is one of the leading causes of preventable death worldwide. Something like 80% of crimes are committed with alcohol involved, violent crimes committed with some kind of alcohol in the mix. Of course, automobile accidents, alcohol is a tremendous contributor to that. And on we go, right? This is a predictable, we have years and years of data that show this is a predictable phenomenon. If we outlawed alcohol, those things would change. Now, we tried that in the United States. Didn't go very well. Familiar with Al Capone, you might be aware of the ramifications of prohibition. But, okay, cannabis also wreaks havoc on the lives of millions of people. People are in prison for life because of possessing cannabis. People are killed because of drug raids and stuff like that because of cannabis, because of prohibition. So right now, the status quo is that cannabis prohibition is causing death and is ruining lives. And it is a tremendous investment by the government into the DEA caring about what happens with cannabis. So frankly, I think that the writing is on the wall with cannabis. It's going to be legalized. I think enough Republicans at this point, Democrats, I think for a long time, not for a very long time, don't get me wrong, but for a bit longer have been sort of on the legalization bandwagon. But more recently, some Republicans now are supportive. Some of the more libertarian-minded, I suppose you might call them, Republicans. And so, yeah, I just think it's sort of the writing's on the wall, and I imagine it's going to be legalized at the federal level. We're guzzling Chimay. Yeah, right. Man, you're making me want to have some of my Chimay, my like 12-year-old at this point Chimay. Are nootropics effective and safe? Great. Thank you for reminding me. So, I mean, here's the short response. There's no evidence that they are effective. There's not a lot of evidence that any particular nootropic is like uniquely dangerous. Doesn't mean that they're not. It just means that there's no evidence that there are. But, you know, I think, you know, the principal question, so for anybody that's unfamiliar, nootropics are a category of supplements that are marketed to improve cognition, to improve brain performance and health. It's a term that was coined, I believe, in the 1970s as a sort of theoretical category of molecules that would be good for the brain. I believe it was coined by a Russian scientist. You know, probably the primary molecules that are associated with nootropics are like paracetam, oxiracetam, and you know there's a whole bunch of racetam molecules, and some of them are used clinically to deal with like ischemia and stuff like that. But they have now for several years been used as nootropics, as like cognition improvers, and there have been studies that evaluate, okay, you know, is paracetam or any of these other molecules, are they actually effective at improving cognition? Do they generate any like measurable change in cognitive performance? And they're with very few exceptions. There's just no evidence that they do. You can find like some studies that will show like an improvement in like verbal recall among a very specific set of you know, the population, like or you know, people who are sleep deprived or who are above the age of 65 or 70, or who are at like a nutritional deficit. Like, so when we're talking about this, that pesky blood-brain barrier, right? So when we're talking about this, you know, I assume we're talking about people who are otherwise healthy, and there's just no evidence to suggest that they are. And the threshold of evidence that it takes for somebody to sell a supplement in the United States is just that they have to not be poisoning you when you're taking it, right? Because if it's difficult to prove that you know, their supplement actually was toxic, but it took like 15 years for it to come about, then they're probably okay. They'll get away with it. But I mean, I can sell you a mysterious white powder and call it Dr. Anthropoid's magical charisma powder, and just put, you know, a little bit of baking soda and some flour and some powdered milk in there, and so long as I'm not damaging you, you can buy it, and I can sell it. There are some really interesting things happening in the supplement world, like oh gosh, of course, there's like a company called Chromadex. There's a company called, oh my gosh, I can't believe, maybe I need some supplements, right? some nootropics. Whatever, a company run by a guy named David Sinclair, a scientist, very accomplished scientist from Australia originally. I think he's at Harvard now, and some folks at MIT, where they're selling a supplement, a longevity supplement, and using the revenue, which is pretty substantial, to fund research into that supplement. That's like a pretty novel strategy. And David Sinclair is like, no, he's like a very accomplished scientist. But that's not really what we're talking about. It's going to torture me. If I don't remember that, it's going to torture me. Mickle Neuro, oh my gosh. Beth Ann, it's great to see you. You will need hardware installed. Mickle Neuro, everybody, is herself a very accomplished neuroscientist, far more accomplished than I am. So we are donuts and the McLaughlins are the best Sunday. Yeah, oh man, making me crave it now. Should we care if our daycare professionals are stoned? I mean, I would, you know, I suppose it really depends upon, you know, the specific circumstances of what the daycare is for, the age of the kids involved, and the reason that somebody might be using cannabis. Not everybody uses Elysium Health. Oh my God, thank you, Beth Ann. Jeez. Yeah, I need some of their Neuro One. It's an interesting story, and I actually covered it on my podcast. It's not all, generally speaking, I'm pretty skeptical of I'm like cautiously optimistically skeptical of the supplement industry, but they're sort of in a class of their own, because they actually do some research, and they actually are some highly accomplished scientists. I think David Sinclair is like the director of the like Harvard Genetics Institute of Aging or something like that, and he collaborates with an equivalently, august scientist at MIT. Mitochondria for sure, and he has studied sirtuins, you might have heard of resveratrol. His lab is one of the first that identified it as being sort of interesting, and there's no evidence that he participated in the falsification of scientific evidence as other scientists have done in the past, so I mean, not everybody consumes cannabis just to get stoned, right? Cannabis, I think at this point, part of the reason that I think the neuroscience community largely is in favor of legalization of marijuana is not only, or cannabis, is not only because there's no good reason for it to be illegal, but there's also, the fact that it has some pretty thoroughly demonstrated therapeutic benefits for, specific populations of people. And so, I think to deprive somebody who's suffering from some kind of malady of a medication that does not damage their body, doesn't make them nauseous, doesn't have unpleasant side effects, to deny them that and deny them employment, I think is unethical. So long as somebody demonstrates competence, I think it's okay. Biden came out for decriminalization. Yeah. That's right. Yeah, I mean, it's great. Politicians change. I first really became familiar with Biden when I was doing a podcast episode on, I think, MDMA, ecstasy, Molly. And Joe Biden was one of the main authors of the RAVE Act. And so, but, who cares? Who cares? Okay. Got to go to school and remove answers from your pockets before you enter. Why do some people have a small brain? Well, by and large, people have about the same size of their brain. But more importantly, the size of the brain really doesn't determine intelligence. I think it's fairly uncontroversial to suggest that humans are the most intelligent animal most of the time. At least we have the most thorough and comprehensive intelligence. But we certainly don't have the biggest brains on Earth, not even close. Elephants have bigger brains than we do, have many, many more neurons than we do. Whales have bigger brains than we do. The secret sauce is in the unique connectivity between the neurons that comprise a human brain, and it seems having this big swollen neocortex that humans have relative to other primates and other animals. While the African bush elephant has something like 256 billion neurons, and we have like 86 billion, our cortex has like 15 billion, and their cortex has far fewer, I think something like 6. It's not all about size when it comes to the brain. There is a connection for sure. Like there seems to be like a threshold, just a number of neurons present to, an intelligence make, but it's not like the determining factor. Ian needs a night with a glass of that poisonous beer. You know what I used to do is I would do have a beer with me, and it would just be a little bit as informal as this is. Believe it or not, it is even less formal. And yeah, that was fun. Now that I have a daughter, and it's just a little bit different. And also, I don't know. I think people, they don't need me to motivate them to indulge. But yeah, you guys are making me crave that heady topper. All right. Yeah, cigarettes, another great example. Yeah. Bonobos are stacking rocks. Bonobos are awesome. I think bonobos are one of the most fascinating animals. We are basically just as closely related to bonobos as we are genetically as we are to chimpanzees. And for a long time, there was this whole model that, chimpanzees are kind of characterized as being far more violent and aggressive and impulsive than bonobos, who generally speaking are less aggressive. And when two troops of bonobos meet, they don't just immediately go to war like chimpanzees generally do. Rather, all the males sort of get together and they do these displays of aggression, but they don't really necessarily fight. And the females go and hang out and groom each other, and then they all have sex. It's a totally different strategy, evolutionary strategy. But so for a long time, I think there was this kind of implied characterization of human intelligence, which was because for a long time it was shown that humans were more closely related to chimpanzees. But and so that might explain why humans can behave so violently, but that's not necessarily the case. Yeah, bonobos are awesome. Talk about the pineal gland. What do you find as its most fascinating attribute? Sure. So, for my PhD research, I focused on a part of the brain called the medial habenula and the interpeduncular nucleus. It's a circuit within the brain, deep buried, basically right in the middle of the brain, that's been in a brain of some kind for over 300 million years. And we know that because fish have it. Name a mammal, they have it. But generally speaking, fish, since they're far enough phylogenetically away from us, far enough evolutionarily removed from us, there can be some pretty meaningful differences in neuroanatomy. But they have it, and so that means it's been around for a very long time, which generally speaking means that the brain structure does something that if you mess with it, if you monkey with it a little too much, it compromises the evolutionary competitiveness of an animal. So it usually means it's an important part of the brain, let's say. Geneticists would hate me saying it that way, but that's basically what that means. So why am I bringing this up right now? Well, for a long time, the medial habenula was thought to literally be derived from the stalk of the pineal gland. Kind of interesting. The pineal gland is a very small part of the brain, and it is sort of anatomically interestingly positioned to suggest that the part of the brain that I studied was somehow evolutionarily related to the pineal gland. That's not necessarily the case. Doesn't have to be the case. But as I said, the pineal gland is a pretty small part of the brain, and were it not for specifically Rick Strassman, who wrote a book called The Spirit Molecule about dimethyltryptamine, or DMT, where he speculated that within the pineal gland, DMT is released, and that is what enables us to have dreams and have near-death experiences. But that was pure speculation. He didn't have any evidence of that. It was just speculation. And he's said that. But after that book came out, the pineal gland took on this cosmic relevance. And it is a cool part of the brain, don't get me wrong, but its primary function seems to be determining circadian rhythms, regulating circadian rhythms in concert with other brain regions like the suprachiasmatic nucleus. Pineal gland is cool. It does receive light information. It's not for vision, but it receives, I think, specifically from certain blue photoreceptors, the prevalence of blue light in your environment, and it is primarily blue light that drives circadian rhythms, which is why a lot of times people who are having trouble sleeping, it's recommended to them that they, either filter out their blue light from their devices, from their screens. There's a lot of apps. I think now it's actually native to iOS and Android, where you can just get rid of blue light, or just avoid devices altogether, obviously. So that's pretty neat, right? Not a lot of brain regions that aren't in the visual cortex receive that kind of information directly. Another really cool thing about the pineal gland is that if you look, I think one of my favorite parts of biology, kinds of biology, is comparative anatomy. And basically, if you look at, a lot of animals have very similar brain anatomy, but there are some meaningful differences. And so, for example, the frontal lobes in the human brain are far bigger than in other primates, certainly other mammals. And by doing that, and then by comparing that to the differences in behavior, in cognition, in capabilities, then you might begin to be able to get a sense of what the function of those brain regions might be. It's just another tool in the toolbox. Okay. So when you do that with the pineal gland or the pineal body, if you look at certain organisms, specifically certain reptiles, it seems to be the case that their pineal gland is high enough, superficial enough on the brain, towards the top enough that actually it functions as a sort of pseudo-eye. It receives light from up above the animal. And there have been some neuroanatomists who have speculated that the pineal gland might have been something like an eye, but just ended up mutating otherwise. So that's neat. But there's no evidence that DMT is released in the pineal gland beyond what's called trace levels. Trace levels meaning very, very, very low levels. So nothing like the concentrations of neurotransmitters with which you're familiar, like dopamine, GABA, norepinephrine, or epinephrine, etc. serotonin. Not even close. And so there's just no evidence that DMT within the brain reaches levels that are, meaningful for consciousness at all. So it's cool, but a lot of brain regions are cool. Like, I personally think the medial lobule is a lot cooler. It's an asymmetric part of the brain that's been there forever. If you inhibit activity in that part of the brain when somebody's withdrawing from alcohol, nicotine, or opioids, you essentially, this is in animal models, can reduce the severity of the withdrawal almost to being undetectable. I think that's a much cooler brain region. The prefrontal cortex, easily one of the coolest brain regions. The ventral striatum, one of my favorite brain regions, the nucleus accumbens. The ventral tegmental area, way cooler. All these brain regions I just named, in my opinion, are cooler than the pineal gland. Are neurons similar to fiber optics? I guess, but in a very small way. I guess they're similar in that information is conveyed between them. Like using fiber optics, you can convey information in the frequency of light bursts, right? So they're a very, very rapid way to convey information. So there's that, and far more rapidly than neurons can. Neurons are actually kind of slow. But that's pretty much where the similarities end. You might have, it's an interesting comparison for sure, and neurons can communicate information by the frequency at which they fire action potentials, or impulses is what I think a lot of people call it. There's some nuance there, but neurochemically through neurotransmitters, and so frequency is important for both fiber optic information communication and neuronal communication, but beyond that, they're pretty different. Now, I think. I don't know if maybe this happened. I'm going to infer that you might have seen some stories about neuroscience that implicate fiber optics in some way, but we're just using I did it in my project, for example. You're just using fiber optics to use, it's a technology called optogenetics, to basically control the activities of very specific neurons in very specific parts of the brain by implanting a fiber optic targeting those regions. But that it's just a tool. It's an interesting. Naked mole rats are the most interesting. Yes, they are. They are very cool. You don't study. You don't work with naked mole rats, do you? Pineal gland photoreceptors, eyeless zebrafish. Yeah, there you go. Eyeless fish. Oh. To see inside the circle while viewing outside the circle simultaneously. Not sure what you mean by that. What happens when neurons all will fire up or get active? So that's why I like to be choked. What happens when neurons all will fire up or get active? So there is this myth, a total zombie myth, that humans can only use 10% of the brain, and it's just not true. We're not limited by anything other the threshold beyond which we would begin having seizures, because it's just uncontrolled firing. And so if let's say Beth Ann McLaughlin, the neuroscientist, mad scientist, found out a way for me to fire all of my neurons all at one time, it would be a very strange experience, but it would be like the most epic seizure of all time. Everything happening all at once, and muscles convulsing, muscles not being able to convulse. Like, it would just be, and perhaps there might be some excitotoxicity in the death of neurons as a result. Mad scientist. Okay. Drug. Okay, so drug you would least like to use because you're afraid of it. Probably intravenous heroin. I think that would be the one. Yeah. Just because like even like the epigenetic changes that occur after just one use, they're pretty meaningful. I would imagine smoked crack cocaine is probably would be pretty Moorish. But just knowing me and knowing how I react to opioids, when I was prescribed, I'm one of those people, the unlucky people in some respects, who is not protected from addiction to opioids, because when I take an opioid, it feels fantastic. I feel, a lot of times people who are prone to addiction, and Bethany, I know you know all of this. This is just for the broader conversation, but one of the sort of classic signs that somebody might become addicted to opioids is if they say something like, when I take oxycodone or when I take heroin or whatever, it's the only time I feel normal. And in fact, was it 2 years ago before I moved, somebody called in, and he had just been prescribed, I think Vicodin, or it might have been OxyContin. OxyContin is oxycodone. I could talk about oxycodone all day. It is a wild history behind that drug, but and he had some kind of surgery. I think he had knee surgery, or maybe it was back surgery. And so back pain is like notoriously difficult to treat, and opioids are not particularly effective at treating chronic pain. But unfortunately, in some cases, that's all that's available. But he called in, and he was just interested in the mechanism by which opioids reduce pain, certain kinds of pain. And the conversation sort of evolved, and then he wanted to talk about the philosophy underlying why we don't allow people to just take opioids whenever they want. And by the way, I mean, I'm for legalization of that. But and he basically said, like, that's what he was saying. Like, I've never felt better or more human, I think is what he said, than when I take these opioids. And that is like a sign that somebody's prone. And I know that feeling. And so I think if I were to ever use intravenous heroin, or really any intravenous opioid, I think I would be pretty hooked. Okay, hey, do you think nutritional deficiencies could cause mental illness? So they certainly can't help. The brain demands a significant proportion of your dietary nutrients, calories. Something like 20% of dietary calories are allocated to the brain. And when it comes to nutrients and minerals, I mean, the brain, if you're not getting enough calcium, and calcium is very critical for the release of neurotransmitters. Essentially, neurotransmitters, almost by and large, will not be released. Serotonin, dopamine will not be released if there's an absence of calcium ions. And fortunately for the brain, there's this gigantic reservoir of calcium, your bones. And if the brain isn't getting enough calcium, it will literally begin to signal the leaching of calcium from your bones to get it. And so, the brain is a very resourceful organ when it comes to its nutrient demands. But for sure, I mean, nutrition, absolutely, nutrient deficiencies will influence things. And so here's, like, generally speaking, the way that manifests. So, as I've mentioned a bunch of neurotransmitters, but also proteins, to be synthesized in the brain, require a series of chemical reactions. In many cases, an enzyme is necessary to catalyze the reaction, and also oftentimes some kind of cofactor, and oftentimes that's what those vitamins and minerals are for. And so, in the absence of those, of like essential amino acids or choline, for example, you won't be able to manufacture nearly enough for the brain to act in the way that it would otherwise. So yeah. Any new research repulsed light for Alzheimer's treatment? I think there was, but I'm not remembering. The latest thing that I read was a study that used, of course, it's like salbutamol or something like that. Essentially a steroid anti-inflammatory that's used for oh my gosh, not for allergies, for asthma, allergies, Alzheimer's disease. And it turned out, I'm pretty sure it was just in a preclinical model that this inhaler, medication for asthma seemed to effectively disrupt the aggregation of the two main aggregates that we think cause Alzheimer's disease. Certainly distinguish Alzheimer's disease from other forms of dementia. The aggregation of amyloid beta plaques on the outsides of neurons, gunking them all up, and the aggregation of neurofibrillary tangles on the insides of neurons, gunking them all up. And so that was a pretty interesting finding, just because essentially nothing has been particularly effective. There have been some studies using like ultrasound to essentially try and literally break amyloid beta plaques apart, and I think there's been some moderate success, or the use of ultrasound to like enhance the ability of various medications to penetrate the blood-brain barrier. But nothing has been particularly encouraging as like a true cure. Even that like asthma medication, salbutamol, I'm pretty sure is what it's called, wouldn't really necessarily even be like a standalone treatment. It would probably be like a complement to other treatments. Okay. So thank you so much for the super hearts, Corinne. I really appreciate it. It's very generous of you all. We are what we eat, indeed. Is it true cocaine used to be given by doctors? Oh, yes. This is one of my favorite topics, but we'll get to that after this. Ladies and gentlemen, we are, I believe, now connected to McLaughlin, a neuroscientist, Beth Ann McLaughlin. What's on your mind?
Second-time caller, long-time listener. Great job. How are you doing in the pandemic?
We're making it through. We're adapting. It's tough because, I have a 1.5-year-old daughter, and, we don't have any like babysitters or anything like that. So my wife and I are just taking shifts, and it's tough. I'm not going to lie. Definitely getting a little stir crazy. How about you? Are you there?
I am doing a lot of reading and just trying to be very patient with myself.
Yeah. Yeah.
I wanted to just put a little aside in there that with neurosurgery, we've been using bubbles to trap chemotherapeutic agents that we then use to quickly disrupt at sites of tumors, and have been very effective. So there's a little bit of promise there, but the blood vessels have to sort of really go right at the tumor, which is usually not a problem considering how highly vascularized they are. But anyway, bubbles. The future is bubbles, Ian.
I'll keep that in mind. That's very cool. Right on.
And I just also want to say, I'm so grateful that you do this. I think this is one of my favorite outreach tools that just humanizes science, and I think everybody should be doing these. You're a tremendous resource, and I'm so grateful for you. So thank you.
That is very generous of you. Thank you so much. And I agree. Frankly, I probably couldn't have finished my PhD if I wasn't able to talk to people outside of neuroscience. So, yeah, awesome. Well, thank you so much for calling. It's great to talk to you again.
Okay, bye-bye.
Bye. Beth Ann McLaughlin. You can find online some of her. She was given a very prestigious award from MIT, and you can find her speech. I think it's if you want to understand what it's like to be a scientist, particularly a female scientist, it's worth checking out. It's just like a very, very beneficial presence in neuroscience. Okay. Oh, sorry, Corinne. I didn't see your topic. So, why do I lose my sight sometimes during a headache? Everything looks like blinking squiggly lines. Wow. Well, so headaches are kind of difficult to understand. There's like different kinds of headaches. Of course, there's headaches that come about by like a brain injury, right? Even, either like right when it happens, you can get a headache, obviously. But then also, recurrent headaches after some kind of traumatic brain injury can occur. That's one bucket. In other words, headaches that are derived from some kind of damage. The other bucket are headaches that come about some other way, and that includes things like cluster headaches, migraines, these types of things. And so there's that, right? And there's a bunch of different potential causes of headaches or of migraines, and some, a bunch of different potential treatments for them that are effective for some people and not for others. Now, what you're describing, so that is a symptom of some kinds of migraines, is like blurred vision, and essentially just distortions of sensory perception. But it's also potentially a manifestation or a byproduct of an epileptic episode, like a seizure. So, at the most basic level, what's happening? The visual system in the human brain is exquisitely complicated, and a tremendous proportion of the human brain is devoted to visual perception, which makes sense. It's a super useful sensory modality, right? As I was saying earlier, it's like it exploits light, which travels at the speed of light, so it's a very quick, rapid, accurate update on what's going on in your environment, right? But as a result, because it's so complicated, there's a bunch of different points along that pathway, from literally the eyeballs all the way back to the visual cortex, which is, again, quite large. And so any kind of disruption along that pathway will impinge upon one's visual perception. And so it could be a seizure occurring in the occipital lobe, somewhere in the visual cortex. And you said blurred lines, right? Squiggly, like blinking squiggly lines. Yeah, so it could be something like that. There's sort of a classic category of people who have seizures that will see, of course I'm forgetting the name, but sort of these kind of glowing, not orbs, but sort of just distorted lines that are sort of sparkly. But there's a bunch of different visual manifestations. They're called auras when people are having a seizure. So it could be that, but it could also just be, not just, but could also alternatively be, a certain kind of migraine. Blotched blank circles, empty. Oh, that's interesting. Well, I mean, the same thing is true regardless of what the specific qualities of the auras or the specific distortions are. But if you haven't brought this up with a physician, you absolutely should. And if they haven't provided any kind of treatment that's effective, it's always worth, if you're in a position, because we have such an insane healthcare system in the United States, if you're in a position to be able to seek, second opinions or just other input from other physicians who might have had more experience with this, it's worth it. Because, what you're describing sounds reasonably common among pretty well characterized conditions. It's just difficult to know which one it might be. Okay, and since those did not understand, they drove a stake through it and turned it into a monument. Wait, what is that? Human being. Let's see, what was your... to see inside the circle, viewing outside the circle simultaneously. Oh, I'm not sure where you're going with that. Sorry, man. Or woman, I guess I don't know. Do drugs help migraine? Yes, absolutely. So once again, we don't know precisely what causes migraines. We have some examples of potential mechanisms that explain some migraines. Like one of them, for example, is, theorized to be the hyperproduction of reactive oxygen species in cells outside of the brain, along the meninges, that then produce inflammatory signaling that's registered as pain by nociceptors, by pain receptors. That's one mechanism by which people develop migraines. And recently, there's a pretty new antibody-based medication for it, in fact, which has been shown in clinical trials to reduce the severity of migraine by 50% in 50% of people, which is, that doesn't sound like, it's nothing to take to the bank, but actually quite literally is something to take to the bank for this company, because that is more effective than, a lot of other migraine treatments. So there's also, there's a bunch more. Actually, there's a, what is it? It's not CREB. It is, I gotta write this down. It's like the fifth time I've forgotten this. Anyways, another molecule that seems to be elevated in people who have migraines, that seems to track with the onset of migraines. And so that's another potential, druggable target. There's a recent paper that just came out that I've been meaning to read that identifies a different molecule. So, migraine, much like many maladies of the human condition, seems to arise from a complicated constellation of various potential causes, only one or some of which one needs to have a migraine. So are there medications for migraine? Absolutely. There's the trypsin-based serotonergic drugs, the drugs that target basically the vasculature along the meninges, that for one reason or another, in some people, they seem, the dilation versus constriction seems to be disrupted, and these drugs like sumatriptan will regulate, I think sumatriptan, whatever, the tryptin drugs, it's a family of drugs, it's not just one, that regulate, because that dilation and constriction is regulated by serotonin signaling, it helps to regulate that and prevent migraines. There's that. There's also psilocybin, believe it or not. Studies have shown that psilocybin at sub-hallucinogenic doses, sub-psychedelic doses, is pretty effective for treating cluster headaches. And that perhaps that's unsurprising given the efficacy of the tryptin-based serotonergic drugs, because, of course, psilocybin, among its pharmacological targets, is serotonin receptors. And so there's that. There's Botox, the use of Botox to treat migraines in some folks. I know it's been effective for at least one person, moderately effective for at least one person, personally. There's Aimovig, that antibody-based treatment that I was talking about. There's a bunch. But what we don't have is a cure, because it's quite likely because it is a byproduct of a variety of activity. I feel like, what were we going to talk about before I started chatting with Beth Ann? Gosh, oh, thank you very much, Jans. I really appreciate it. Or Jan, maybe. I appreciate it. It's very generous of you, particularly these days. I really do. Yeah, microdose. We can talk about microdosing if you'd like. Actually, I was going to do a whole podcast episode about microdosing because there is some research now. Just so everybody's on the same page, microdosing refers to microdosing. It refers to the use of, generally speaking, psychedelic hallucinogens like LSD or psilocybin or mescaline or some of the more obscure and esoteric psychedelic molecules that were invented by Alexander Shulgin, like the halogenated amphetamines, for example. But generally speaking, people seem to gravitate most towards LSD, but it's using them in minuscule amounts. So generally, LSD can be dosed in a variety of ways. It can be dosed most classically on blotter paper, basically like little stamps, essentially, that are absorbent paper. And because LSD is active at the microgram level, it's like incredibly potent. All you need is just what's in a drop that's dropped on a piece of paper, and then you put that on your tongue or under your tongue or something, and that's enough to have an effect. But then, if you even, take just a fraction of that, that's what microdosing is referring to. The short story is I wouldn't recommend it just because the pharmacologies of these drugs are so versatile, are so broad, and the effects on states of consciousness of the activities of psychedelics can be rather unpredictable. But I'm not 100%, I'm cautiously optimistic that there's something there. What I would hope is that but I just wouldn't recommend it because it's not thoroughly evaluated. And I think, philosophically, here's why. Like, there's this famous, I think, Swiss scientist named Paracelsus, who is considered the father of modern toxicology. And he has this quote, I'm going to butcher it. It was probably not in English, but the thing that distinguishes a poison from a cure is the dose. And so, the doses that people associate with psilocybin or LSD are just determined by the fact that they've always been associated with psychedelia or psychedelic hallucinogens. But that doesn't mean that is the beginning and the end of their pharmacological potential utility. So I'm not against it, but I just want to see more evidence before I do it. The point of financial incentive will eventually cease. I'm not sure. Microdose? I'm not sure what you mean by that. Confluence of triggers. Is it true that doctors used to prescribe cocaine? Oh my gosh, it's one of my favorite topics. Thank you so much for reminding me. Space Ghost. Dojo. Great choice in Avatar, by the way. So yes, I mean, the short answer to that is absolutely. In fact, I've been really wanting to purchase you can get bottles of like Bayer, Bayer branded cocaine for toothaches, for nausea, for a bunch of stuff. Thanks a lot for the super high, man. And it's wild for coughing. I think it was even prescribed for children. And in fact, some of the most influential physicians in the history of medicine were severe cocaine addicts. In fact, Johns Hopkins University, which really was like the institution that was pivotal to standardizing high levels of medical care and standardizing the types of medical care and like treating it like less of an art and more of like at least basing it on science. Johns Hopkins was pivotal. And then so was the University of Pennsylvania. And in fact, I used to live in the building where a lot of those students almost certainly were educated, which is very cool. It was turned into a bunch of apartments. It was Graduate Hospital. I lived in the Graduate Hospital. That's a neighborhood in Philly. Anyways, one of the founding fathers, there's the four founding fathers of Johns Hopkins. I think his name is William something with an S. And he was a notorious cocaine addict. But he was fortunate because there just so happened at the time to be the perfect treatment for cocaine addiction, which was heroin, also sold by Bayer. And so yeah, he became I'm pretty sure heroin, not morphine, but either way, it was I'm sure he became addicted to that. Yeah, and in fact, there's a TV show. It's one of my favorite shows of all time called The Nick, which is sort of based off of this guy, one of the founding fathers of Johns Hopkins. It's with Clive Owen in the show, and it's really good. I totally recommend it, particularly if you're interested in this kind of thing. It's not just about cocaine, although that does play a pretty significant role because a lot of doctors use it. Notoriously, Sigmund Freud used it, right? But it's also about like the inception of the use of gloves, or the invention of the C-section, cesarean section, or the use of cocaine as a surgical anesthetic. Pretty cool show. I highly recommend it. What's it called? It's called The Nick. Think like the Knickerbocker, the Knickerbocker was like a hospital that at the time like the turn of this last century catered towards lower income people in New York City. Yeah, William S. is played by Strother Martin. William Sonoma. William, I can't remember. How long can a person stay? Oh, you know what? What time is it? How long has it been? Let's do that. That should tell me. Why is it not telling me? Okay, sorry. This is not awkward, I promise. Oh, wow. I've missed so many topics. Okay, let's go through these. Oh, no. Okay, it's just some sort of pleasant discussion. Yeah, I've wanted to get for myself. Gosh, I can get rid of this. I wanted to get one of those bottles, those like antique bottles of cocaine, for years, and I just haven't done it. I bet I can get it on eBay. But yeah, you can even get like there are, I think at Penn actually, in like they have like little mini museums as you walk through the campus, particularly the biomedical parts. There are like little vials of like morphine from like 1902. Which I think is just absolutely fabulous. Anyways, I should probably call it a day. Thank you so much for watching. I really appreciate it. I hope it's as beneficial for you as it is for me, keeping me sane, keeping me from totally losing my mind in this pandemic. Wear a mask. Keep wearing masks no matter what. Keep social distancing. Something like every 150 Americans has this thing, or has had it at least. And I have a YouTube channel where earlier I talked about the almost 8% of your genome that is derived entirely from viruses. So not from like other animals, evolution, but from viruses specifically. My YouTube channel is youtube.com/anthropoid, or you can just search Ian Brain Anthropoid or something like that. Really cool story. I also talked about viruses that are so huge that they begin to blur the lines between what scientists consider to be living and what scientists consider to be not living. And they're called gyruses. Literally, they're called giant viruses. But I have some pictures that you can check out. Really cool story. I'll probably talk about them in greater depth later at another time. And I have a podcast called Wired to be Weird. Talk about almost every single one of the topics that came up today. I've talked about in greater depth on the podcast, excuse me. So Wired to be Weird on iTunes and Stitcher. And finally, I know everybody's in a tough spot these days because of this damn pandemic, but if you're in a position to be able to contribute, I have a Patreon, patreon.com/anthropoid. I only ever use that funding for getting more equipment, and the next thing I need to get is a fixed microphone stand because this one broke. But no matter what, I want to keep doing this as long as I can, regardless of whether or not people can support. But I appreciate it if you can. And so with that, I'll let you enjoy the rest of your Sunday. Thank you so much for joining, and I will see you same time, same place next week. See you there. All right, YouTube. Thanks again for watching, everybody. Again, if you have any input, I'm super receptive. And if you haven't, I would appreciate it if you would subscribe. Smash it. Smash that subscription button. Anyways, thanks for watching. Have a great weekend. There's my daughter.